Hi, my name is Catriana. I'm with the Chow Hub at the University of Pennsylvania and Children's Hospital of Philadelphia, and I'll be talking about modeling thrombocytopenia absent radius syndrome or TAR syndrome using induced pluripotent stem cells. So, so thrombocytopenia absent radius syndrome or TAR syndrome is a rare congenital disorder that's primarily characterized by bilateral absence of the radii while maintaining the presence of the thumbs, a severely reduced number of megakaryocytes or MEGs, and thrombocytopenia, which is low platelet count. And patients with TAR can also present with a number of other different phenotypes like cardiac abnormalities, some renal anomalies, as well as a cow's milk intolerance. It's caused by a combination of a deletion in one allele of the RBMA gene, which results in a null allele, in addition to a single nucleotide polymorphism, and typically a non-coding region of the other allele. And the most common of which is in the 5' UTR. And this results in reduced RBMA mRNA expression, as well as significantly lower levels of the encoded protein Y14 in patient platelets. But currently, Y14 has no known roles in megakaryopoiesis. So this study aims to understand how the deficiency in this protein leads to the megakaryocyte or MEG phenotype that's seen in TAR patients. So one mechanism by which Y14 might be affecting megakaryopoiesis is through its role as a core factor in the exon junction complex. And the exon junction complex is typically placed onto mRNAs by the spliceosome just upstream of exon-exon junctions during splicing, and it remains bound until it's removed during translation. And this allows it to impact many aspects of the mRNA life cycle, including mRNA transport, surveillance, translation, nonsense-mediated decay, and of course, splicing. And so both depletion and overexpression of EJC core proteins have been shown to cause a wide range of alternative splicing changes, including exon skipping, alternative 5' or 3' splice sites, and intron retention, which demonstrates a clear role for the EJC in regulating alternative splicing. And when Y14 is depleted, many of the genes that demonstrate alternative splicing changes are involved in processes that are known to be regulated through alternatively spliced isoforms, such as apoptosis and cell cycle regulation. So for example, Y14 knockdown by siRNA in HeLa cells demonstrates a switch in the splicing of genes such as BCLXL, MCL1, and P53 to produce isoforms with more pro-apoptotic activity and leads to increased cell death. Additionally, Y14 depletion has been shown to cause abnormal mitotic phenotypes, decreased mitotic regulators like CDC20 and CDK1, and cell cycle arrest, which is all indicative of defective cell cycle progression. And in further support of this, a more recent mouse model of TAR that knocked out RBMA specifically in megakaryocytes demonstrated a blockade of MEG differentiation due to a cell cycle disruption and expression of pro-apoptotic isoforms of P53, which suggests that there's some sort of regulatory network between Y14 and P53, which might be contributing to the MEG phenotype. However, this was also a complete knockout of RBMA, which is known to be lethal, and it might have been causing P53 because of the total knockout. And it neglects the low level of RBMA expression that's usually still present in TAR patients. It is also well known that there's many biological differences between mice and humans, especially megakaryocytes. So it's important to model these diseases in humans in addition to the mice as well to determine if these defects are conserved. And so during development, megakaryocytes and other blood cells are first produced after around 3 weeks post-conception in the yolk sac of the developing embryo. Here, early mesoderm cells differentiate into more hematopoietic or hemangioblasts-like cells that then bud off hematopoietic progenitor cells. These primitive hematopoietic progenitor cells, or HPCs, will then mature into primarily nucleated erythrocytes with some primitive macrophages and megakaryocytes. But to become megakaryocytes, these progenitors will make a series of lineage decisions to distinguish between the other cell lineages until it's committed to the MEG fate. And so they undergo first polyploidization, in which several cycles of replication without cell division results in a huge, massive polyploid cell that can accumulate a DNA content ranging from 2N up to 128N. And the megakaryocyte then matures its cytoplasmic contents by developing a demarcated membrane system to serve as a membrane reservoir for subsequent platelet production and producing alpha granules. They then shuttle these packaged platelet contents in those alpha granules through long cytoplasmic extensions called proplatelets that will extend through the vasculature to be sheared off by blood flow and then mature into platelets. And so, in order to provide the majority of pre-mRNA, spliced mRNAs, proteins, and regulatory machinery for RNA processing that's been found in anucleate platelets, these MEGs have to greatly increase their RNA and protein synthesis. And in order to survive such a very stressful maturation process, MEGs depend pretty heavily on the regulation of apoptotic factors, specifically like BCL-XL and MCL1 for survival. And so, given that the MEG maturation process seems to depend on the correct, efficient splicing of proteins involved in cell cycle regulation, RNA processing, and apoptosis for proper maturation, they may be more sensitive to changes in the splicing of these proteins due to EJC core depletion. So, we therefore hypothesize that Y14 expression in TAR syndrome alters alternative splicing in megakaryocytes, and that this change in splicing contributes to the MEG phenotype that we see in TAR patients. And so, in order to address this hypothesis, the GDU lab has generated two patient-derived iPSC lines that both contain the 200 kilobyte deletion and one allele of RBMA, as well as a single nucleotide polymorphism in the 5' untranslated region of the other allele of RBMA. And in addition to the patient lines, we also generated corrected lines that successfully restore RBMA expression in TAR hematopoietic progenitor cells and subsequent megakaryocytes using a gene targeting strategy that inserts a CD43 promoter driven copy of RBMA into the AAVS1 locus. And as you can see here by a CD43 GFP reporter line, the construct that CD43 driven will start to express as the hematopoietic progenitor cells begin budding off of this hemogenic endothelium-like layer, and will express whatever gene of interest, in this case GFP, and in our disease model, RBMA, and all subsequent hematopoietic lineages, especially megakaryocytes, as you can see here. And so for our case, starting at this stage where the progenitors are budding off, we should see a corrected or restored expression of RBMA in our patient lines. And so once we generated these iPSCs to generate hematopoietic progenitor cells, we use an established primitive differentiation protocol from the GDU lab to produce these hematopoietic progenitor cells, or HPCs, that can be used for modeling hematological diseases. So it's designed to kind of mimic that primitive hematopoiesis that we see in vivo with different combinations of cytokines to induce various lineage decisions made through development, like mesoderm, more endothelium-like mesoderm, until it's the mangioblast-like layer that buds off these primitive progenitors. And so we can get this nice CD41, CD235 double positive progenitor cells in order to differentiate them into megakaryocytes. So to do this, we put them in a media mix with thrombopoietin, one of the main drivers of megakaryopoiesis, as well as stem cell factor to expand progenitors. And once we get these megakaryocytes, there are several ways that we'll measure their maturity and functional capacity. So for maturation assays, they're often assessed by staining for the expression of MEG-specific cell surface markers like platelet glycoproteins like CD41 and CD42B or CD61, and measuring these levels with flow cytometry. And a higher mean fluorescence intensity correlates to higher maturity, and higher percentages of these mature cells in the population can correlate to more mature cell culture populations. So you can also use these values to calculate the number of mature megakaryocytes that are produced from a starting progenitor population, kind of as a readout of cell expansion and differentiation capacity as well. And we also assess the functional capacity of these in vitro derived MEGs through agonist responsiveness and proplatelet forming capacity. So in platelet agonist responsiveness assays, they detect changes in the conformation of the surface platelet glycoprotein 2A, 2B receptor, which is critical for platelet aggregation in order to allow a PAC1 antibody to bind as an indicator of activation. So the MEGs are stained with MEG-specific markers like CD41 and 42B, as well as PAC1, and then treated with an activator like convulsin or thrombin, and measuring the PAC1 binding subsequent to activation. And so a higher percentage of PAC1 binding upon stimulation correlates to more functional megakaryocytes. And then to predict the proplatelet or platelet forming capacity, proplatelet formation assays quantify the percentage of megakaryocytes that attach and form at least one proplatelet-like structure in response to a fibrinogen-coated surface. And so a higher percentage of these proplatelet forming cells correlates to a higher functionality in that population of megakaryocytes. And so this is where I currently am in this project, and some recent differentiations are yielding pretty promising results. But the next steps of this project would be to determine if TAR causes changes in alternative splicing. So I'll purify these megakaryocytes with FACS sorting, and perform RNA-seq on each condition. And so I'll be focusing on genes pertaining to MEG development and proplatelet formation, as well as the cell cycle and apoptosis genes that I was mentioning earlier. And so differences in these genes would identify potential mechanisms for any megakaryocyte-specific phenotypes that I would see during the maturation and functional assays, and support the hypothesis that the TAR phenotype might be due to changes in alternative splicing as a result of this Y14 depletion. And so there are also a number of future directions and areas for significance for this project. So for one, as more definitive fetal or adult-like hematopoietic differentiation protocols are optimized and developed, it'd be interesting to repeat these experiments to determine if these changes are conserved in a more mature or more adult-like population, or if they change due to developmental timing. And in doing these experiments, it will help us to improve more disease models in order to kind of understand how different diseases where the cause of thrombocytopenia is unknown may be due to changes in alternative splicing of MEG-specific genes. And if we can better understand how splicing of relevant genes or how to produce more mature functional megakaryocytes, we can develop better differentiation systems for ex vivo megakaryocyte and platelet generation. So that would be able to generate a higher yield of platelets for subsequent patient transfusion, for example, and more translational downstream applications. And so just as a summary, the results of these experiments will expand our understanding of how alternative splicing of relevant megakaryocyte genes might affect biology, or in order to develop more accurate in vitro disease models or generate a more sufficient number of platelets for transfusion medicine. And so I would like to acknowledge the Good Lab at Penn, and also the Human Embryonic Stem Cell Core, CHOP. And many of my projects are supported in part by the Developmental Biology Training Grant, or T32. And I will now open the floor for any questions.